Good afternoon. Today we're going to learn about photosynthesis, and specifically we want to know, how does photosynthesis transform light energy into stored chemical energy? So does anyone have a partial answer for this question? How about you, Ryder? What do you think? You're saying the sun? Okay, well the sun is the major energy source for most plants, but this light doesn't automatically get transferred into energy. It must first go through a chemical process, which is what we are going to learn about in this lesson. So we're going to move on to autotrophs and heterotrophs. All life on Earth depends on the flow of energy. We talked about this yesterday in our lesson. And remember that this happens throughout the ecosystem. The source of this energy is the sun. So, the sun fuels life on Earth, and beyond that, all organisms fit into one of two categories. They are either an autotroph or a heterotroph. Remember that auto means self, and hetero means different. So for autotrophs, those are organisms that can make their own food. These organisms use the light energy from the sun to produce food in the form of glucose or sugar. This includes all green plants, some bacteria, and some produce. Heterotrophs. These are organisms that cannot make their own food. Examples are all fungi and all animals. Remember that heterotrophs must consume food. Therefore, they eat plants or they eat other animals that eat plants. So you're getting a system where you start with plants, and then you have animals eating those plants, and then other animals eating smaller animals, and it goes throughout an entire food web and a whole ecosystem. Energy enters the ecosystem in the form of sunlight. Plants use the sun's energy to make glucose. The sun's energy is stored in the molecule of glucose, and the energy moves up the food chain when a consumer eats the plant. So, if you see this picture of the food web, it shows you how energy gets distributed. You start with the sun's rays go down and they're absorbed by the plant leaf. Water comes up through the stems, or through the roots into the stem, and this gets converted into glucose. Here's a molecule of glucose on the side. Then insects will eat the plant, larger animals like a frog will eat the insect, and finally you move all the way up to predators such as a hawk eating the smaller animals. Remember, photosynthesis is converting radiant energy from the sun into chemical energy in the form of glucose. Chemical energy and ATP. Inside living cells, energy can be stored in chemical compounds. One of the principal chemical compounds that cells use to store and release energy is ADP or ATP. ATP is adenosine triphosphate, and ADP is adenosine diphosphate. ADP is energy poor, but when one more phosphate is attached to the end of the molecule, it's converted into ATP. So ADP has two phosphates. You attach another phosphate group, and you get an energy-rich source like a charged battery. The structure of ATP. It consists of an adenine, which is this orange compound over on the side. It's a nitrogen base. A ribosome in pink, which is a 5-carbon sugar, and a chain of 3 phosphate groups. ATP has enough stored energy to power a variety of cellular activities, such as photosynthesis, protein synthesis, muscle contraction, and active transport across the cell membrane. The ATP molecule is the basic energy source of all living cells. In a cell, ATP is used continuously and must be regenerated continuously. In a working muscle cell, 10 million ATP are consumed and regenerated per second. Here's an overview of photosynthesis. In photosynthesis, plants use the energy of the sun to convert water and carbon dioxide into high-energy sugar molecules. Oxygen is then given off as a waste product. Life on Earth is dependent on photosynthesis for food and oxygen. So you can see the sunlight being absorbed by the plant. Right here, the sun energy is going down. And you're getting water from the ground coming up through the roots. Carbon dioxide is coming in through the atmosphere, and they're all combining inside of the leaf and having a chemical reaction. That reaction combines together to create a sugar, and then oxygen is released as a byproduct. The photosynthesis equation. This is the equation right here. You have carbon dioxide and water and sunlight all mixed together to create sugar with a release of extra oxygen. Photosynthesis uses the energy of sunlight to convert water and carbon dioxide into high-energy sugars, which are glucose, and then oxygen. The carbon dioxide is found in the atmosphere and is taken in by the leaves of the plant. Happens here in the stomata. The water is in the ground and is absorbed by the roots of the plants. Plants make their own food through photosynthesis. So here's another illustration of this process. Again, sunlight is coming down. It's being absorbed by the photoreceptors on the leaf. Water is coming up through the roots, through the ground, and carbon dioxide is coming into the leaves through the atmosphere. These are combined together to produce a sugar, and then oxygen is released as a byproduct. So you have sunlight, carbon dioxide, and water turn into sugars. The sugar is stored in the plant, and you also have oxygen, which is released into the atmosphere. Remember that leaves are the major organs of photosynthesis. None of this can happen without the special properties of the leaf. There are about 500,000 chloroplasts per square millimeter of leaf surface. So this is all happening in these little tiny factories. They're like chemical reactors that are called chloroplasts. And we are going to get further in depth with this as we go on. We're going to learn how chloroplasts collect light in our next lesson. Right now, you need to know that photosynthesis consists of both light-dependent reactions as well as dark reactions, such as the Calvin cycle. But it does get pretty in-depth, and we're not going to have enough time to cover that today. Now we're going to move on to the water dilemma. Plants do have a problem with this process. The major problem that happens is that they face dehydration. Plants must open their stomata to let in the carbon dioxide that is required for photosynthesis. But anytime the stomata are open, there will be excess water loss through the stomata. In order to prevent water loss from the plant, plants are covered with a waxy cuticle. There will have to be trade-offs or compromises between photosynthesis and the prevention of excess water loss. So that's why plants aren't always optimal with photosynthesis, because if they did as much photosynthesis as possible, then they would dehydrate themselves. So they need to just kind of curtail that system a little bit by producing waxy surfaces on their leaves. It does stop the amount of sunlight that can be produced, but it keeps in a lot of the moisture at the same time. Temperature is also a very big factor in this process. It depends on the action of enzymes. Now we know that enzymes work the best at temperatures between 0 degrees Celsius and 35 degrees Celsius. That's right around close to 70 degrees Fahrenheit, which is the perfect temperature for humans, so plants are not unlike other animals. Temperatures above or below this range may damage the enzymes and prevent them from functioning. At very low or high temperatures, photosynthesis may stop entirely. And then you have light intensity. We know that increasing the light intensity increases the rate of photosynthesis. So that is why you get a lot of plants in the equator zone, right where you get the most amount of sunlight. You have perfect conditions for plants to proliferate, and you get a lot of plant activities. All right, to sum it all up, the energy from the sun has to be stored as chemical energy in glucose. Do you want to answer any questions that you have? So we are going to go ahead and start our Elode Photosynthesis Lab. The objective of this lab is to see a visual representation of photosynthesis in the classroom. The Elode plant will be able to fix the water and CO2 in the test tube to make sugar and oxygen. The oxygen will be produced underwater, creating bubbles, which you will be able to count. This lab will also show the importance of light as a factor for photosynthesis. So, our initial question right here, what is the relationship between light intensity and the rate of photosynthesis in this Elode plant? You have a section right here for your hypothesis. You're going to get together with your partner and think about what is the best answer to this question. What do you think is going to happen between the light intensity and the rate of photosynthesis? Then you can gather your materials. On every lab table you have an Elode plant stem, a test tube, distilled water, sodium bicarbonate, scissors, a light source, and a timekeeper. You can go ahead and use your cell phone if you would like, or a watch. So the first part of the procedure is to set up the experiment. You're going to take your plant, remove several leaves from the bottom, cut a portion of the stem at an angle, and lightly crush the cut end of the stem into the test tube. Then place a small pinch of sodium bicarbonate, that's on everyone's tables, into the test tube. This is going to increase the carbon dioxide in the water. Then you're going to place the plant into the test tube, bottom end up. Fill the test tube with distilled water so that the stem is completely submersed. Now the next part, when you're ready to begin this experiment, you're going to place a source of light about 5 centimeters from the plant. Everyone has a lamp, heat source right next to them. After you do that, you wait 1 minute, and then you count the amount of bubbles of oxygen rising from the cut end of the stem for 3 minutes. Remember, it has to be exactly 3 minutes. Keep the tube in the light. If bubbles fail to appear after 2 minutes, repeat part A. Then you're going to run a second 3-minute trial at 10 centimeters from the light source. Then you record your results for this trial. Then you run a third trial 3 minutes with a distance of 15 centimeters from the light and record your results. You're going to record your results on this table right here in the middle. Distance from the light source, 5 centimeters. You're going to count the number of bubbles that you see in 3 minutes. Then you do it from 10 centimeters. You count the number of bubbles again. And then finally from 15 centimeters, you're going to count the number of bubbles that you see. After you do that, you prepare a graph of your results. It's going to be down at the bottom of the page. Your graph is going to be on this square right here. The x-axis will be the distance from light, and the y-axis will be the number of bubbles that you see in 3 minutes. When we're done, we're going to compare our results, talk about what you think was happening, and then turn them in. So right now you can go ahead and begin your experiment with your partner. Remember to first record your hypothesis. It's really important. I'm going to walk around and assist you and answer any questions that you have. Thank you.